He's a manager of the biotechnology and the biochemistry labs. He has a degree in agronomy with a specialization in plant breeding and genetic resources. He obtained a PhD in 1997 on genomics of coffee trees and an accreditation to direct research in 2017. His current researches are on the optimization of genetic selection, including major QTLs in different type of populations for plant morphogenesis and quality in the context of changing climate and low input. He is in charging and leading Work Package 5 in Euclid. Philippe, please, you have more than 1 hour for your presentation.
So thank you, Dejan. So I will give a presentation. Yeah. On genotyping-by-sequencing, GBS, a sample method for a multitude of markers. And I prepared this presentation with Tom Rutting, who is here, and is going to present something later, but if we have questions, we can discuss together. My presentation will be in 3 parts. The first one, I will give the general principle of genotyping by sequencing, how it works. Then I will go a little more deeply in how to make a library, and then how to analyze the sequences. Okay. So first, what I mean by genotyping. So it's getting the genotypic classes or the allele frequencies at different loci of a sample. So if you have a locus, you want to know either if it's AA, for example, or AB or BB. And you can say also you want to know if it's 0, 0.5, or 1. So this is for diploid plants, but it could be also for a population. You want to know the allele frequency, and you will look at the frequency of A. So that's genotyping. You want a table with all the samples and this genotype information. And with the dramatic decrease here of the price of sequencing, it has been possible to use sequencing for genotyping. Here you have the price of getting a complete human genome sequence. And you see it was $100 million here, and it's now about $1,000. So you see that this dramatic drop of the price allows the fact that we can use sequencing to genotype the different samples. So before it was, okay, you have one human and you do the sequencing on this one and you genotype the others. And now the idea is to get access to 100 human and to sequence all of them, and you get their genotype. So, okay, it's possible, and the price has decreased a lot. But even though, if you have a lot of money, you can do whole genome sequencing. And for, like I said, it's $1,000 for a human, but it's about half of it for alfalfa, which is quite expensive. So if you look at the price of sequencing, it's too much in comparison to the value that you will get with this information in comparison to phenotyping. And, but for some species, so here I put flax, because I was involved in a project in flax. And here on this species, you see the genome is small, and it has been possible to do the genotyping by whole genome sequencing. And so we did GWAS analysis, and like 400 plants were sequenced completely. Just one point here. You see, I didn't say sequence here. I say resequence or resequence. Why? Because by that I mean, first of all, you need a good quality sequence. And it's much more expensive because you need to get the sequence without any errors. But in addition to that, you need to build the sequence and to put together, to assemble all the little part of the sequence to get a good value sequence. And for that, you need different technique of sequencing. You need short sequences with good quality, and you need more longer sequences with maybe a less good quality, but that allow you to put together, to assemble the sequence. So here, when I say we can do the whole genome sequencing, I mean resequencing. And it's not the same price if you do it the first time. So in the majority of cases, it won't be possible to do whole genome sequencing. So we will have to reduce, and usually they say to reduce the complexity of the genome, meaning we won't sequence everything, we will sequence only a part of the genome. Which part? So you can decide, and it has been done several times, to focus on genes. And for example, you will sequence RNA message mRNA. To do that, you need to select the plant material that you are going to extract mRNA, and you know it will depend on the physiological stages, and so you can get only a part of information if you focus, for example, in mature leaves, and you don't get buds or stuff like that. So this is convenient, but you can get only part of the genes by doing that. So what is usually done is to pick several samples from one plant, and so we will have a little bit of flowers, a little bit of leaves, and at different developmental stages. So that's one way. Another way is to target some specific loci. And this can be done with amplicon sequencing or by capture. So by PCR, amplicon sequencing. And the other capture, so you have probes and you cut your DNA, and with probes you select the DNA you want to sequence, and with that you extract it and you do the sequencing. To do that, you need some information on the sequence you want to look at. So if it's the first time you work on a species, it won't be possible. You first need to have sequence information to design the primers or probes. And you need the sequence information for, often we can have it for one sample, but it's not enough, especially with forage species. Because with forage species, the variability that you have in the species, it's just huge. You have a lot of variability. You have one polymorphism, like one SNP, every maybe 15 base pair. So when you design your primer, you have a lot of chance to design the primer where you have a polymorphism. So it won't work for all the samples. So it's important, before starting that, to get good information on the variability you have in your species, and especially the variability you want to survey in your breeding program, for example. If you use primers from a different study, maybe half of them won't work in your own study. So that's something important, to have an idea of the variability before starting this kind of targeting specific loci. Okay? So if you know nothing, one option is random sequences. And for that, you can use restriction enzymes to to cut your genome. And so this has been published by Elshire and collaborator in 2011, so with a robust sample genotyping by sequencing approach for high diversity species. But even though, so this is the basis, oops. This is the basis, I think it's an article where you have all the information in GBS, so the basis of GBS. But even though there is patents, because you have several patents from Kijin, from the Netherlands, where, no, I think it's better like this. That's, yeah, life, I confirm it's weird. So you have patents from Kijin for the SNP identification by using restriction enzymes and high-throughput sequencing methods. It means that if you want to perform GBS, you will have to buy a license. Or you have the possibility, I put here, to work with, in Europe, you have the LGC group, and so they provide the service to get GBS analysis. So now I will go for the general principle of GBS. So the first step, so now we are looking at, we want to reduce the diversity of the genome, and we want to sequence only part of the genome. So the first step is to digest the genome. So it's here, it's, this is the genomic DNA for one sample, sample one. And you digest here, and you have the restriction enzyme sites in here, and you have one SNP here from this sample one, and in comparison to this sample two, where you have an A and a G. So you digest, and you have many different sizes. If you look at the distribution, you will have a nice distribution with a different size of the different fragments. And you can't do anything with that right now. It's just you have that. So the idea, and this is, okay, was the idea of Kijin several years ago, was to put adapters. So adapters is non-sequence. It's a short non-sequence, DNA sequence, like at least 18 or 20 bases. And since you know those sequences here, so you ligate the adapters on both sides of all the fragments. And the idea is now it's not just unknown fragments. It's fragments with non-adapters. That was the idea. Pretty smart, I think. But then, since you know the adapters, since you know the sequence, you can use it to do PCR amplification, because you can design the primers knowing the adapters. One point here that I want to mention is the fact you see you have a little blue stuff here, and it's a little thing, so it's kind of star, and it's green there. This is the barcode, which is a little bit of sequence, a very short sequence, and this is unique. It's a sequence unique that you don't find anywhere. And so this barcode will be specific to each sample. So you have a blue barcode here and a green barcode. And this is important because with that, even now, if I put together, if I mix the sample 1 with the sample 2, I will be able to recognize sample 1 and sample 2 thanks to the barcode. So, okay, we have now fragments with non-adapters, and we can do a PCR with that. And so the amplification, here you have the first selection of your fragments, because the amplification won't work with very long fragments. So you will only have fragments between, let's say, 50 to 450 base pair. So you have a selection of the fragments. So you only get some fragments that you will be able to sequence afterward. And here I put between sample 1 and sample 2, you see you have a loss of restriction site. Before, we were using this information of loss of restriction sites to have what they say AFLP markers, amplified fragment length polymorphism markers. So if you make the amplification and you run that on a GL, you have different bands, and you can have different of sizes because of loss of restriction enzyme. And that was AFLP markers at that time. And it was also patented by Kijin. And now the idea, it's not to look at those polymorphic sites with restriction enzyme, but to focus on sites where you keep the restriction sites, so you can amplify that, and we will look inside the sequence and see if we find SNPs. So the fourth step is sequencing. Should have had, before sequencing, you put everything together, all the samples together. So then you do the sequencing, and now you have a bunch of sequences, a lot, like, I don't know, several million reads, short reads. And okay, it's a big file. So what you want to do is to, first, you want to figure out where are your samples. And this is done with a software or several software by looking at the barcodes. And that's what we call the demultiplexing. So demultiplexing, it's you have all the sequences in one file, and then after demultiplexing, you will have several files, and in each one, it's for one sample. So you find sample 1, sample 2, sample 3, etc. And here, so what you got, it's a FASTQ file, and it's like that. You have a header here, then you have the sequence, and then here the FFF and 2 points and stuff, it's the quality. So you have an information on the quality of the sequence. And this can be used then to filter out some bad sequences. But again, it's a quite large file. So this is demultiplexing. So now you have, for example, if you treat 200 samples at a time, you now have 200 files to analyze. So the next step will be to clean your sequences. Because like you see here, so you have one. Here, the first one is only blue, so it's only the fragment that you have because you sequence. And it's a short sequence, or it was a long fragment. You didn't reach the adapter. So you sequence only the fragment. You didn't sequence the adapter. So it's already quite clean. You can clean it for the quality, like saying that if the quality is too bad, I don't want to keep this sequence, but this is okay. Then, for the two others, you see, for the first one here, the sequence was short, so the sequences went until the adapters. And so you have a little bit of the adapters in your sequence, and you don't want that for the further analysis. So you clean that. And you get something like this, many of that. So next step will be, okay, I have maybe 10 million clean sequences, and I'd like to know where are they in the genome. So you make an alignment by homology. So the sequences, you align them against a reference sequence. So you have a sequence from the reference, and you align. So you look at the homology, and you say, okay, this one fits here, this one fits here, etc. And you see for one locus here, you have several reads on that. That's what we call the read depth. And I put another word here, coverage. And coverage, it's when you look at where you have sequences along or across the genome. And often you have confusion with coverage and read depth. It's difficult, yeah. So coverage, it's along the genome, and read depth will be at one locus, how many reads you have. And what you get, it's a file. So the first file will be a .sam file. It's an alignment file. And because they are still quite big, you can reduce, you can have files which are compressed and with indexes. And so this will be a BAM file. So often you use the BAM file. And here you have an example of a SAM file. So you have the name of the sequence. You have the name of the reference that you use. So here for us it was the Medicago sativa sequence that we got in the EUCLEG project, and it's the pseudomolecule or the scaffold here 127, and you have the position where the read starts, so the position here, for example. And then here, it's the Siger code alignment. And this tells you, okay, your sequence is along the reference sequence without a stop. That's what it says here. For 88 base, M means it's following the reference, but it can be a I or a S, depending if you have an insertion, for example, or a deletion. So it tells you the correspondence between the read sequence that you have here and the reference sequence. So at each position, you will know, okay, the read sequence is that, and the reference genome is that. So next step will be to say, okay, now I have all my sequences, my reads, align on the reference sequence, and I want to know where I have polymorphism, because I am interested in polymorphism. So polymorphism will be variant. And often, here I said, we will focus on biallelic SNP, like A, G, or something else. You also have tri or tetraallelic SNP, but often we focus on the biallelic SNP. And another thing is that you also have the information of insertion and deletion, but here also we focus on SNP because it's difficult to treat afterward, and also because the insertion deletion could be wrong, the position could be wrong in the alignment. So when you look at the sequence here, reference sequence and the reads, you see only one SNP at position 452, and here you see you have one like the reference, it's orange, and 4 like the alternative, it's red. So the variant calling will say in sample 1, at position 452, the reference it's a A, the alternative it's a J, and we have one reference, 4 alternative. That's the number of reads. And then you have a big step here because it interprets that and give you the genotype. And here it say it's a GG, or you can say it's a 00. So here you have an interpretation of the number of reads. And this has to be carefully done with a geneticist to know what kind of information, how do you interpret, when do you decide it's homozygous. Here you see I have 1 against 4, and I decide it's a GG, it's homozygous. I could have, when do you, if I have 2 and 3, do I decide it's heterozygous or not? Do I have enough information? So this has to be very clear and discuss with the geneticist. You can check several thresholds. Like I said, here you see I have only a read depth of 5. Is it enough? I don't know. What is your... Because here it's really, this is a decision that we have to make. We have some reads. For you, when you have 5 reads, who is going to call for SNP here with 5 reads in the assembly? I don't know if... So it's a diploid. You have 5. Do you think it's enough information to say it's homozygous, heterozygous? Who would call for a variant with 5 reads in the room? So you can raise your hand. So, yeah, you call for a... Yeah. So the other, they don't want to... No, you wouldn't. No, okay. In fact, when you look at publications, when you have the information, because sometimes you don't have the information, you have people with 5. For a diploid, it's not a problem. They would call. Even with 2, sometimes. Yes, that's true. I saw it in publications. But we have to be very aware that it's a decision there. And the results that you get depends dramatically on that. So this has to be discussed with the geneticist and has to be checked afterward with some segregation and some family and stuff. For me, with 5, it's not a problem if it's homozygous plant. For that, it's no problem. when you have heterozygous, when do you stop? 2, 3, it's heterozygous, right? I wouldn't do that. I would say, okay, I prefer to get a little more. Like 8, 10 maybe is good, it's better, to be sure that it's heterozygous. So I insist on that because it's the beginning, but it's very, very important to be sure. And often you don't see it, in fact. When you can apply a software on your data, and it's by default, you don't see. So you want to ask and be sure what the software is doing.
So your suggestion is to like 80?
Yeah, I think it's a kind of probability. And so for me, yes, 8 would be, you can decide. But then when do you put the threshold? So you have also something to decide.
Yeah, so if it will be like 5 to 3, you think
Yes.
it's heterozygous, it's 6 to 2?
Yeah. So what we usually do, in fact, it's we take all the information, the raw data, so you know the number of reads, so you have the frequency. You can calculate the frequency, and you make an histogram of the frequency, and you see if you have a continuous histogram like that. I don't know what to do. If you have like here a lot of homozygous 0, and then something like this, and this, and this, okay, I can say here, I will cut. And I kept some security, and I say from 0 to 0.22 maybe, it's homozygous 0. And then from 0.3 to 0.65, it's heterozygous, like this. But if you don't see, I check that.
Yeah, okay, thanks.
Okay? Another thing important here, and just if you do the variant calling, the information you get out of the variant calling, it's only this. Sample 1, there is a polymorphism at position 452. That's it. But you see that when you do that, you lose the information that here you have enough reads to call, but it's monomorphic. But you lose this information for this sample. But maybe in another sample, you will have a different allele here, but you won't know it. It will be a missing data for this one. So it could be better, I think it's better, to keep the information for all loci where you have the information. And you can do that with the getting a file called GVCF. So instead of looking just at where do you have polymorphism, you keep track of, okay, I have reads there, and I know it's like the reference. But I know it's like the reference. It's not like here. Here, for example, it's missing data. I don't have any reads. So you make a difference between missing data and homozygous reference. This is also something important, I think. The kind of file you get then, it's a VCF file. So you have the chromosome, like the reference sequence, the position on the reference sequence. This is the reference allele, the alternative allele. And then here you have 1,1, it's the interpretation of the genotype. So it's homozygous alternative. The 187, 115, it's the number, so it's heterozygous. It's the number of each reference and alternative, and then you have the total number of reads. So you see in this VCF file, you have all the information, you have the interpretation that you can use, if you trust it, if you have checked it's working, and you also have the raw data, you know how many reads for reference and alternative. And this is also something you can use, and you can make your own interpretation then. And this is a GVCF, and you see it because it's positioned, you see here, 48, and then it's 49. So you keep track of all the information everywhere. It's nice. It's costly in memory. So you still have big files, and you have big files. Yes, just here I wanted to be sure to give an example for the difference between the VCF and the GVCF. And for example, in sample 1, I put what kind of data I will get in the VCF. So here I will have missing data. There is no polymorphism, missing data. Here, that's what we saw. We have a SNP and then missing data because there is no reads. Missing data, missing data. In the GVCF, here you will have, so if I take one position here, you will have 6 reference, 0 alternative, like 5 reference, 0 alternative, 1. And this is important because here, look, on sample 2, it's 5 alternative. So in fact, this SNP is fixed in sample 1 and sample 2, but it's polymorphic. It's not the same allele in sample 1 and sample 2. If you just do the variant calling and get only the variant here with the VCF, you lose this information. So depending on the software you're going to use, be very careful with that. So now you have all the GVCF file for the different samples. What you want to do is to merge all this information in a single file, so you will be, that's what you want at the end. At the end, you want the SNPs, samples, and the information, so for all the samples. And that's what is done here with the merge variant calling file. And so you have the chromosome, the position, reference, alternative, and now you have the information of the genotype, and you still have the information of the number of read of alternative and reference, but this is for sample 1, then sample 2, sample 3, sample 4, etc. So you merge all the information. And that's where, when you don't do a GVCF and you just do the VCF, you will have missing data for all the reference homozygous sequence. So with this file, in fact, you have the information you want. You have the 0001 for all the samples. So it's like this. You have the SNP 1, SNP 2, and for sample 1, it will be 0, 1, missing data, 0.5. So you're ready for the analysis, almost. If you use it like that, you will get a lot you will get some problems because of the missing data. So you have to do a filtering. And this is usually done, so you take this file, it's a text file. All the file I showed, it's text file, so you can open it with whatever. So if you like R, you can use it and open it with R, and you filter, check for missing data. Per SNP, but also per sample. So that was the principle, general principle of GBS, and this is a summary. So library preparation, digestion, ligation of adapters, amplification, sequencing, and then the bioinformatics with demultiplexing, cleaning, alignment of the reference sequence, variant calling, merge variant calling, and filtering. And I put here again to be done or by or with a geneticist to check when do you decide. But you have to check that. So that was on the principle of GBS. Do you have question on the principle? Then I will go a little deeper in library preparation and bioinformatics. But is there any question on the global principle, or is it clear?
Yes, there is something that I don't understand in this step number 7 and 8 with variant calling. I don't understand why you can have doubt whether a locus is homozygous or heterozygous. How can you, if it's homozygous, how can you have a different variant in these short sequences? Maybe it's good to go back to the slide. Yeah,
Like in this one I can show?
Yeah, one before, I think. There, you were saying that it's difficult to interpret whether you have to do with a homozygous or a heterozygous species individual, and I do not understand why there is doubt. If you have two different variants, why is it not clear?
Because okay. Here, for example, here it's I put 5 only reads in purpose because 5 it's just a few. But for example, now I said I have a read depth of 30. So I have 30 reads and I have 1 reference. What do you think of this value? In fact, it's a sequencing error.
Yeah, okay.
And so you don't know where to put the threshold of the sequencing error.
Okay. So these differences can be due to sequencing errors?
Or other errors. Okay. But it's your background. And you want to, you want to put the threshold, where is your background? When you think it's not a background anymore.
Oh, yeah. So, and can you use a chi-square test to determine whether this is by chance or by not by chance? Is this helpful there?
These things are incorporated in the software.
Yes.
Okay.
All of these interpretations are automatically incorporated in the software. It's shown here in a sort of simplified way, but there's a lot of statistics going on between the read depth and the actual interpretation. So there are many different forms of variant callers. Some of them use Bayesian statistics, others use other types of statistics, but there's always a distribution of read depth behind it, and a certain chance to call it as a homozygous, a certain chance to call it as a heterozygous.
Okay, but now I understand from where this comes.
So I agree, but you don't know exactly, often you don't know exactly what the software is doing, and when you check, you can't check everything, but you pick some SNPs and you check, and sometimes it's you say no, it's not possible. How? So for example, GATK, which is a software, how could it say there is a heterozygous there? I don't trust that. So often when you look at the distribution and you see a drop and then it goes, here you really see your threshold, and this something I trust. But for that, you need not 5. But in publications, they use the software, and you can find sometimes it's just with 2. And they claim that, in fact, it doesn't really matter at the end because you have so many markers that even with 2, you make errors. Even with a lot of missing data, you make errors, but you have so many information, you see the signal afterward.
I have one maybe stupid question. You said that it is quite demanding in memory. Do we need some supercomputer for computing that, or?
Yeah.
So computers.
Yes. You need both, in fact, but I will come back on that a little later. But you need 2 things. You need hard disk memory, so a lot of memory because you have big files and you compress and decompress, and so you produce new files. So you need a lot of memory, and in addition to that, you need a lot of CPU, so computational power to do all the work. You want to complete?
Well, there's a difference between a supercomputer and a server.
Yeah, I was thinking of a server.
A server is enough.
Yeah. Yes. Yeah.
One more thing about this. You said that you can decide, of course, with the help of a geneticist, for example. So is that a choice depending on the kind of species you're working on? Or, I mean, your material? Because you said that, for instance, if you have just 3 rib depth, but with homozygous or heterozygous, you refer to what in that case?
If you have autogamous species.
Oh, so it's okay.
If you have an autogamous species, you
so
don't
crossing or
yes, you don't have to differentiate the heterozygous, so you can drop a lot the number of reads. Yeah, maybe another one there.
It's quite a detail, but I had a question about the cleaning step. Can you show the...
Yes.
So you had one read with no barcode, and you said it's, well, you keep it because it's already clean, but how can you be sure of the demultiplexing?
So yes, yeah, okay. This step is one step further. So first, you do the demultiplexing and you get that. And so even for this one, we add the information with the barcode. And during the demultiplexing, we threw it away. And often, in fact, this is made by the sequence provider.
Okay.
But you're right. You have to know which barcode do you have here. That's right.
Okay.
And it was the step after. Yeah.
Thank you.
At the time. So, shall we continue? So, library preparation. So, I took some slides from Elshire and all, and I want to go through the slides with you. And so the idea presented in this article that, okay, you can do you have a plate with many samples, so 96 samples there, and you will digest DNA separately, ligate within each wall, and then you pull the 96 DNA aliquot. So in this article, in fact, the idea is that it's the fact that you can do the digestion ligation within each separately, so this is not too expensive, and then you pull everything. And then you have one step here, which is cleanup, and this is a bit expensive, but you only do one for 96. So that's the thinking. And so here you clean, and you got this, and then you have the PCR that you do on one. So it's also a bit expensive, but you do it on all together. And again, you clean, but it's on the pool and all together. Here, PCR, you do several PCR to avoid if one PCR is not working well or not. And then you check the size. So this is here. It gives you the size of the fragment, and these two, here and here, it's standard where you know the size, and the library is in the middle here, and you check the size. So that's very convenient, and that's very cheap. But sometimes what you want is to come back to the samples, and for some experiment, we do the PCR for each sample separately so that we can go back to the PCR to resequence if we want. But the cheapest way of doing it is the way presented by Elshire. So now, if you look at the adapters, it's like that. So, like I said, it's sequences, it's DNA sequences, and you have the Illumina sequencing primer, so the primer 1 and primer 2. You have the sticky ends for the enzymes, so it will recognize the restriction site and stick there, and the barcode, and here it's 4 to 8 base pair. And you can use different enzymes, like here you see APK1, PST1, etc. So then, when you do the PCR, you don't use just the adapter as the primers. What you use is this here. So you recognize the P1 and P2, but in addition to that, you see O1 and O2. And this is a sequence that will be used for sequencing. So it's here. So you have this after the ligation, and then after the PCR, you got something like that, with the insert in black in the middle. Here I put something. I said it's essential to have links between the lab, who is doing the library preparation, the lab who is doing the sequencing, and the person in charge of the bioinformatic. Why is it so essential? Because depending on what kind of sequencer you're using, you have certain constraints on the design of the barcode, for example, because they don't use exactly the same fluorochrome. They don't use exactly, so when you do the sequencing, you have different way to do it, and depending on the sequencer, you have to take this, you won't have the same barcode. So this is essential that you have a good communication between the person in charge of the library preparation, because it's nice, you can do it in your lab, it's just pipetting, you don't need a robot for that. Then the sequencing, often the sequencing is done in a specific lab because the sequencers are very expensive and they update all the time, so it's not possible to do it in a small lab, but you have to talk. And then you get the sequence back and you can do the bioinformatic in-house, but again, you need to know what has been done and how the libraries were prepared. So here it's sequencing using Illumina ISEC 2000. And so this is the principle. You have here kind of, I would say, a slide or something. They call it a flow cell. And here on the flow cell, you recognize the O1 and O2 put on the flow cell. And so you will stick your library, the different fragments, on the flow cell. Then you multiply this, because if you only have one, you won't see the signal. So you have to multiply this one, like this. And then you will do a sequencing, sequence synthesis of that. Here, you see in this drawing, it's nice. You have well-separated fragments. You have one here, one here. This has to be very precise, and so you have to know exactly the amount you're putting on your flow cell, because if they stick together, if you put too many, you won't be able to sequence, because it's, you won't see anything. If you put too less, you will be able to sequence, but you're losing money, because it's very expensive. So this is something which has to be done by a specialist. And then you have the sequencing process. So for example, here with the primer, sequencing primer number 1 in blue, and you start, in fact, you have a synthesis of your, the DNA fragment. And when you do the synthesis, in fact, you use fluorochrome with different colors. And so each time, for example, here you have a T coming, it will give you a red signal that you can see here. So this is a picture of the flow cell, and you see the signal. So each time you see, if it's, for example, a red, it will be a T, and then a yellow or blue or green. And this kind of techniques, and the fact that with that you're able to sequence a lot of reads at the same time, this is the drop of the price. Because of this new technology, compared to what was done before, now it's possible to do it for a lot cheaper. Here you have so the sequence forward in this way, but you can also have a sequence here in the other side, and so this is paired end sequences. So you can imagine here that if you put too many fragments at the beginning, all the little spots are clustered together, and it's not possible to sequence. Here it's just to give you an example of what we are doing in the lab. So it's just to show you that it's, when I say, before that it was just, it's DNA. So here you have the sequence. It's actually a sequence. So you can order this. It's a sequence, and you order single-strand DNA, like primers, and then you can make your own adapters. Here you see the barcode. Here this is the other adapter, and here we used PST1 as a restriction enzyme. And in this slide, you see something else that I didn't present yet. It's the fact that when you want to analyze a lot of samples at the same time, you won't have enough barcodes. So what you can do, it's to use here what they call index, so index 5, or on the other side, index 7, and it's the same principle. It's a little bit of sequence that is specific to the sample, and by doing that, so we use it in Coxfoot, we use 29 barcodes, 5 index 7 and 2 index 5, and with that we can put together 290 samples at a time. And here I put again have to be compatible. This is important, and this has to be discussed with the people in charge of the sequencing. And this we put on the sequencer. So the sequencer that we are using now, it's in Toulouse, and it's a Novasec. So it's last technology of sequencer. And we got about 4 billion of reads for about 11,000 euro. And so we get something in the average 13 billion reads. No, it's a 6 here. We get 13 million reads per sample. And so we have from 2 to 25 million reads per sample. So you see here, we got from 2 to 25. This is not good. 2 to 25 million. I want something like 10 million. So here, we have to do again some samples. That's why I said before, yeah, sometimes it's better to do it separately, even if it's a little more costly, then you can pick the sample again and redo only those ones for resequencing. So you have to check the quality of the library. You look at the size of the fragment, and here you see some dimer of primers. And so you can decrease that by optimizing the amount of adapter or the cycle of the PCR. And you can also clean with different protocol. Now, give you some information about the choice of the restriction enzyme and the number of reads. So this is from an article from Byrne and al., 2013. And you have here, it's on perennial regress, and this is with APK1. So it's quite frequent cutter compared to PST1 here. In the axis, you have the number of reads in million, and in the y-axis, you have the number of loci. And for example, here, if I take a read depth of 20 in red, you see that for about 20 million reads, you will get something like 200,000 loci. But you also see that here, in fact, the potential number of loci that you can get with APK1, it's much more than that. So in fact, so you have some loci with reads, but in fact, you don't have enough reads to put information on all the loci here and here. So at the end, if you do something like that, you will get a lot of missing data because you didn't get enough reads to cover all the loci where you have APK1 restriction enzymes. If I compare that to PST1, you see the curve here, it's kind of reach a kind of plateau. The slope decreases slowly. And here, if I take 20 million reads, I got something like 50,000 loci, which is a lot less, but for the same amount of reads, so when I say for the same amount of reads, you translate by for the same amount of money, you get 50,000 reads, but without too many missing data. So depending on what you want to do, the number of markers you need, if, for example, if you are doing a QTL analysis in a population where linkage disequilibrium is very high, you don't need a lot of markers. So you want to choose restriction enzymes that cut not that frequently. If you want to do GWAS in perennial regress, where linkage disequilibrium drop after 150 base pair, you need a lot of markers. Then you change and you choose another restriction enzyme that cuts highly frequently, but it has a cost. So you need to calculate that and to estimate, and depending what you want to do, if you have a certain budget, it's better to say, okay, I will get less markers, but at least I won't get too many missing data, than to say, okay, I try with APK1, I will take the threshold at 2 reads, and I will call a variant. This is, you will get many missing data, and I think it's not that good. In some cases, you can do that. And if you look at what people in Cornell do, in fact, they have a lot of missing data. But they know exactly the genotypes, so this is in maize, they know exactly the genotypes they are working on it, they are working with. They have the sequence of the ancestral genotype. And because of that, even if they have many missing data, they can infer the genotype where you have missing data. But this is a lot of work behind and a lot of information used. So most of the time, it's not the case. So you have to think, what restriction enzyme do you want to use? And here, I put 2 examples, one in Dactylis and one in alfalfa. And you see we used different, it's not only one enzyme, but it's a couple of enzymes. And for example, for Dactylis, the population we're working with, it's a kind of mapping population with quite long linkage disequilibrium. So we don't need a lot of markers. And so eventually we pick this one. You see, we reach a plateau quite quickly, and we can get 10,000 loci with 10 million reads. And with alfalfa, we picked PST1, MSE1, and it's about the same. So the choice of a restriction enzyme is essential. What can you do with GBS that you can use with populations is the fact that you can estimate the allele frequency of a population. And this is something that often you can't do with other markers. So this is an example with perennial regress also. And here it's the allele frequency estimated based on 48 individuals. So each individual were treated separately. The SNP were obtained per individual, so DNA extraction per individual, SNP polymorphism per individual, and with that you can, with 48, you can calculate the frequency of each SNP based on the individual. And here in X, you have the allele frequency, but estimated on a pool of leaves from 48 individuals. And you see that you have quite a good correlation between the pool of leaves. So you estimate directly allele frequency on a pool in comparison to do it 48 times, because you have to do it per individual. And so GBS for us in fresh species is essential because we can do that. We can estimate allele frequency on pool of leaves. And this here, it's so in X it doesn't change, so a pool of leaves, and here it's pool of DNA from the individual. And here also you have a very good correlation. What you have to know is that the estimation is good, but you see the prediction interval is about plus or minus 0.12. So it's, you can differentiate between 0.2 and 0.6, but 0.2 and 0.25, it's not possible. But still, it's very convenient, and it allows us to study many populations. Before that, we were doing individuals separately. So 40 individuals per population, it's too expensive. With that, it's possible. So with that, study on alfalfa, so Julie, Bernadette, and all, we are done to look at the diversity and to look at the possibility to distinguish between varieties. And so with 20 varieties, you see here, one from Estonia, one from Canada. This is specialist in turf, grazing, Germany, and those varieties were from France. And when you look now in the variety from France, you see that you have the possibility to distinguish between the south and the north. And even in the north variety, we can distinguish between the breeders. Here, so Geogras, Després, Barenbroek. Before that, with SSR markers, we tried with AFLP, and in fact, the FST, so the differentiation between the varieties, is quite low. It's like 0.05, and it was not possible to distinguish them. With GBS markers, since you have like 10,000 markers, it's possible to distinguish between alfalfa variety. And I don't know, yeah, some of you work with alfalfa, and you know the kind of diversity you can have in one alfalfa variety. So this was something very useful. And yeah, and I hope that's something that we will be able to use to have new varieties with good value that we couldn't distinguish. So I think that's it for the library preparation. And where I am in time? I'm late. 10 minutes. So no questions. We'll see later. So bioinformatics. Bioinformatics, the idea, first, you see what I put here, it's a multitude of softwares and on Linux. And a multitude of softwares, so you have quite a lot. Some are more used than others, but you have quite a lot of software. I put here a little something. Possibility to use Windows. So for example, I used CLC Genomics. It worked perfectly to find, to do the demultiplexing, to do the alignment, to do the variant coding. Nice, quick. The only thing, the problem I got is you can do it for samples separately, and when you want to merge, you lose the information of the fixed SNP on the reference. It's not possible to have the GVCF. So that was, for me, a major limitation of this, but you can use it for a different study, and it's very convenient because you see your sequences. It could have a limitation when you have a lot of samples to deal with. So now, what we do, it's mainly with Linux. And so for the first, the demultiplexing, often it's performed by the sequences provider, and you can use so different software, Sabre, GBS6, and others. And we tried GBS6, and it worked fine, so it's not a problem. The cleaning also, you have you want to remove the adapters, so you have different software like a CutAdapt or a Site. You want to remove bad quality reads or part of reads because of the size, so you have another one, it's a Cycle. Removes short reads, it's Cycle. So you have different software that you can use, and so often you have some of them that you liked better than others, but you have to figure out which one, and you have to test, and you have to look what you have at the end, and if you are happy with that or not. And here I put so one file. It's 10 with 10 million reads. It's about 2.5 gigabytes without compression. So necessity of a large hard disk. For the alignment, it's the same. You have quite a bunch of servers. Like I think the most used is BWA, so ALN or MEM, but you have others. And here I took this from the article of Elisabeth Wickman, and she compared BWA and GSNAP, two different software for mapping reads. And so you see in dark gray, it's the same when you get the same SNP with both software, but you see that you have a little bit of light gray and dark, and this is specific SNP from BWA or GNSNAP. So depending on the software you use, you have quite a lot of common SNPs, but still some of them are different. So maybe you want to do something with that. Usually you want to compare. So at least you have to be aware of that. And it's the same with variant calling. I think it's worse with variant calling. So when you have different variant caller like GATK, Freebase, or others, and here again in the article of Elisabeth Wickman, she compared different variant callers, and here you have, so this one, it's when you have the same SNP in all the variant caller. And so in black here, it's about 33%. So when you compare these four variant colors, you have 33% of the SNP that are in common. Okay, this is the way it is. But often people don't realize that. So I think it's here what I want to say, it's just be aware of what you're doing when you're using this software. If you just, okay, I use BWA and GATK, and you don't check what you have, it will work, it always works. But you can have false positive. And when you do that, and you get, for example, you do a GWAS, and you find a SNP very nice, and you say, oh, this one is it's wonderful, it's explained a lot, and it's highly significant. Go back to the raw data to be sure that there is everything right, and it's you trust it. Yeah, I think that was my message for this. And yes, in addition, yeah, you can have a look at this article. I think it's nice. It's a variant metacaller. And the idea here is to apply different variant colors on the same data, and in fact, to put together all the information in order to pick the trustable SNPs. And here I put the necessity of many CPU and large RAM to do that, so informatic stuff. So for a new software I've been made is in progress right now and will be proposed soon. So it's from Tom, who is right there. It's SMAP for stack mapping anchor point. And the idea here, it's when I explained to you, we will focus only on SNP. You know, you remember at the beginning I said, we now forget about the differences of amplification. We focus on the loci where everybody is amplified. But you can have some differences because of insertion, deletion, or other stuff. And the idea here is to be able to point out and to get out the information and use it as new markers. And in addition to that, you have the possibility to have a haplotype. So I think I should go a little faster there. It's the end, huh? So SMAP will be presented a little bit maybe tomorrow or this afternoon if you want. It's a possibility. And so just my conclusions for this topic, GBS. So GBS could provide a multitude of markers, that's true. Don't forget that the markers, they are not targeted. So once you have SNPs that you want to follow, for example, because you have QTLs there, you have to change the technique. So, okay, it's very good to look at the diversity at the first step, but then maybe you want to change and look at other markers. For a relatively low cost, so you saw in one of my slides, you saw a cost, so it was to give you an idea. But you have to be aware of the patent. So this, I don't know the cost in LGC. Yeah, we can, you can ask. It's could be done for any species. So this is something I didn't talk about it, but you saw that I said that you need a reference sequence. In one of the slides, you will see it's not always a necessity. You can work without a reference sequence. And in fact, what you do is to use all the sequence that you have from the GBS library, to use it and to build a reference, and then you map on that. And there is an article with an example on sugarcane. It's on the slide. Without too many missing data, so I hope I convince you that this is true, but you have to be careful on the choice of the restriction enzyme in adequation with the budget you have. And relatively quickly, and I said here, with a good sequence provider and a bioinformatic team or pipeline. And this is also something, we do the bioinformatic in-house, but this is also something that the sequence provider can provide, the bioinformatic, and give you the genotype. But please, again, if you do that, ask how they interpret their read numbers. You want to know exactly what they do with that. And on that, I think I thank you very much for your attention.